Lastly, we need to make a distinction between compendial and non-compendial methods. Both in US and in Europe, compendial methods need to be verified before initiating phase 1 clinical trials. So as regards non-compendial methods, in US, for initiating phase 1 clinical trials, you need to have qualified your dose-determining methods as well as safety methods. This way, it is ensured that the product is safe. When moving towards pivotal clinical trials in the US, dose-determining methods need to be validated, and all the rest of the release methods need to be qualified. However, a validation plan should be ready beforehand. When approaching commercialization in the US, all the methods should be validated. That means release methods, in-process control methods, and also stability methods. These should all be validated for a BLA. So meanwhile, in the European Union, before initiating first-in-human clinical trials, all methods need to be suitable for purpose before initiating phase 1 trials. This approach reflects the EU stringent standards that focus on quality assurance right from the very early stages of development. EMA recommends to prioritize validation, meaning that first you will need to validate your safety, dose-determining methods, potency methods, and then stability indicating methods. So both in Europe and the US, ICH Q2 should be followed for method validation, and you need to provide details regarding the validation protocols, meaning the samples, positive and negative controls, reference lots, equipment, all that stuff, and of course data supporting the validation. So in early stages of development, it is not needed to submit protocol or validation reports, but for later stages, they should be included in the submission.